Hi, everyone. If you're joining us a little bit early, we're going to give it a minute or so until we feel like we're ready to start here with regards to attendees. Thank you. All right, I think we're there. So we're going to get things kicked off, everyone. Thanks for being here and welcome to our 24th episode of Ewings U. For those of you that don't know me, I'm Piero Spada and I'll be your host today. I'm also co-founder at Little Warrior Foundation and more importantly, father to now 14-year-old little warrior Lucy. And if you're part of our core contingency, AKA the Little Warrior Fight Club, thanks for being here with us since day one and making this a place for positive change. And if you're new, well, welcome to Ewings U. It's our webinar series where we hear from leading sarcoma experts and discuss the cutting edge of care and science behind Ewing sarcoma. Today, we'll be hearing from one prominent expert and Dr. Bernadette Brennan to discuss a new clinical trial called INTER-EWING-1. Briefly, Professor Brennan is a consultant pediatric oncologist at the Royal Manchester Children's Hospital with over 30 years experience as a qualified doctor, as well as further training in general pediatrics, pediatric and AYA oncology, and an MD in late effects of childhood leukemia. She focuses on Ewing sarcoma, osteosarcoma, pediatric soft tissue sarcomas, and rare pediatric tumors. She is a national coordinator for the non-rhabdomyosarcoma soft tissue sarcoma study, aka the NRSTS, run by the European Pediatric Soft Tissue Sarcoma Study Group, otherwise known as the EPSSG, and chair of the NCRI Sarcoma Group. She is an active member of the Euro-Ewing Consortium, including as chief investigator of the UK Euro-Ewing 2012 study, principal investigator for the RECURL trial, and chief investigator of the INTER-EWING-1 trial, the subject of today's webinar. She is also a member of the Children's Cancer and Leukemia Group, aka the CCLG, and she is the CCLG's rare tumor group, part of that, and the expert European rare tumor group. No shortage of things you're involved in, Dr. Bernadette Brennan. But before we get things kicked off with Dr. Brennan, just a quick reminder that Ewings U is a webinar production of the Little Warrior Foundation. We are a national childhood cancer foundation with a laser focus on finding a complete and permanent cure for Ewing sarcoma. We were founded in April of 2020 and have now granted out over $2 million in high potential therapies for increasing survivorship and decreasing toxicity. And in order for us at Little Warrior Foundation to make programs like this a reality, we rely on the support of the little and big warrior community alike. We appreciate all of your support so that we can continue our mission of bulldozing our way towards more effective and less toxic therapies and continue this vital work for kids battling Ewing sarcoma. With that, I'm going to hand it off to our omnipresent Emily to touch on a couple of housekeeping items.
Thanks, Piero. Yes, I'm Emily McFadden, one of the other co-founders of the foundation and aunt to Little Warrior Lucy. We are excited to kick things off with Dr. Brennan, but first, a few housekeeping notes. We anticipate that there'll be a lot of questions today, so please use that quick Q&A functionality in the bottom of the Zoom chat. We'll save the questions for the end, but please send them through at any time. So also, we can't discuss specific cases on this webinar, so please keep the specifics out of the Q&A questions. So with that said, we will pass it over to Dr. Brennan to get started.
Okay, thank you very much for inviting me to do this talk to your organization, Little Warriors. And so what I've been asked to talk about is I'm going to go through, I've called it INTER-EWING update, but it's actually just to go through the current trial, which we're running, I'll show you where we're running it, but it's mainly Europe with Australia, New Zealand, and it's for newly diagnosed patients with Ewing sarcoma, and all stages are included in this one study. This just gives you who all the group are involved. So it's based at the University of Birmingham in the centre of the UK, and there's a Children's Cancer Clinical Trials Unit there, which is core-funded by the big cancer charity in the UK, Cancer Research UK, and Cancer Research UK also have funded a large component of the study. And then there's various trial coordinators. If you're involved in the study, you'll get to know about. And this just gives you a map of the participating countries. New Zealand and Australia are new partners into the European Ewing Consortium. So we're not just European anymore. Obviously, the United Kingdom, and the original study that was the forerunner to this was Euro-Ewings 2012. That was United Kingdom, France, Ireland, and Spain. But subsequently, this study, which, if you like, is the follow-on study to that, includes other countries in Europe, such as Denmark. Italy's been a big partner. We brought in Norway, Poland, Switzerland, the Netherlands, and Czech Republic. So they're the sort of blue areas on the European map. There's always been another study group based in Germany who have run separate, since about 2008, have run separate Ewing's trials. So unfortunately, we're not all Europe is in this study, but we're probably the larger of the studies. So some of this background, a lot of you will already know, but most of you will know that Ewing sarcoma is a small malignant tumor of bone and soft tissue, and it frequently affects children, adolescents, and young adults, although there are some slightly older patients who get it. And most importantly, this is an all-age study, so it's children, young adults, adolescents, and adults. From the last study, we established that VDC-IEVC is the standard of care, and this was a European-based study. And obviously, VDC-IEVC is standard in United States through the COG group, but we compared that with what was then the European standard. So instead of giving chemotherapy every 2 weeks, as you do in VDC-IE, we used to give it every 3 weeks. It was like all the drugs together. That basically showed that not only was VDC-IEVC superior by about 10% in terms of outcomes, but also was actually less time in hospital and less toxic. Also, what's important, whether you've got localized or metastatic disease, is local control, surgery, hopefully, plus or minus radiotherapy, but some patients have definitive radiotherapy. So I've done some of my screen. What have I done with my screen? Yeah, here we go. So we know that if you take real world data as opposed to specific trial data, the event-free survival, if you take all patients, is between 60 and 75%. But obviously, some patients with widely metastatic disease, their survival is really down towards about 20%, 25%. And there were important things when we started to think about questions for this study. So this is all newly diagnosed patients with Ewing sarcoma. So none of these patients previously had been treated. What we regard as standard of care, particularly for radiotherapy, as our local therapy, whether it's with or without surgery, there's actually very little evidence to support what we think is the standard treatment, either in the dose that we use and how many fractions we give. And there's never been any randomized studies looking at different doses to the primary tumor site, whether it's after surgery, before surgery, or as definitive radiotherapy. And you'll see lots of different doses being used across internationally, not just in Europe, but across the States. The other thing was that there's never been any maintenance treatment after the end of the typical 14 courses of treatment of VDCI-EVC. And there is some evidence for maintenance treatment in other sarcomas, such as rhabdomyosarcoma. And so, again, we wanted to ask the question whether if you add maintenance therapy, whether that improves outcome. Trying to get it to move now. Sorry, my slides. Is it moved? Can anyone see? Is it the same slide?
It's the same slide. I don't know if you can try using your arrows.
Yeah. Might be. Let me just see. Might be the screen it's in. I might just reshare. I don't know why now this isn't working. Let me just see. Just give me a minute.
We've all been there. I don't know why it's. Let me just see if that works now. Right, sorry. Right, that's working. Okay, sorry about that. Dr.
The
Brennan.
trial.
Oh, no, now we see it. Okay, perfect. Thank you.
Is that okay? Yeah.
That's great.
So,
Yep.
so this is a phase 3 clinical trial, and our primary aim is to improve the outcomes of newly diagnosed Ewing sarcoma of all stages. And it actually is in a very efficient trial because it has multiple randomized questions at multiple time points in the treatment pathway, as well as collecting other information. So no patient data is wasted at any point. So it's all newly diagnosed patients, and they should, where possible, enter the trial at diagnosis, so prior to any chemotherapy. But patients can actually enter the trial at any point of the randomization, but ideally you want them to be admitted at the beginning to the trial, register on the study, and then they can be randomized at various time points or not. They also are providing consent for biological samples, imaging data, the obvious clinical data, and all the treatment details. And as long as you have the sort of standard of care VDCIE chemotherapy, then you can enter at any point, even though you may not be eligible for a randomization up front. There are certain countries in Europe who like to use high-dose therapy with busulfan melphalan, either because they've got poor-risk localized disease or metastatic disease. And this is very much a European phenomenon. It doesn't tend to be practiced in the States. And so that is permissive. It's not part of the trial question, but because it's a randomized study, so any question will hopefully have an equal spread of patients who've had high-dose therapy or not in each arm. And we'll follow patients up for a minimum of 3 years from the point of last randomization or until death if sooner. So the questions, the first question is a chemotherapy question. So the first question is, if you give an extra drug, which is regorafenib in this study, in addition to the standard treatment, do you improve survival where the disease has already spread? So this is for about just over a quarter of the population will either have widely metastatic disease or metastases just to the lung. And then the second chemotherapy question is how long should we give it for? So this is about adding in a maintenance treatment. I'll come to this. And again, trying to improve survival. Then we have randomized radiotherapy questions. So there are some patients who cannot have any form of surgery other than a biopsy at diagnosis. And so in that scenario, we're going to compare two radiotherapy doses, one of which for some it would be regarded as a higher dose in this scenario. And also where you are wanting to give post-operative radiotherapy, either because the tumor was large or has a poor response to chemotherapy. And so in a lot of European Ewing studies, we use a factor of necrosis, so the percentage of the tumor that's dead. There has been a definitely comparison of different doses of chemotherapy, radiotherapy, from what would be regarded as a lower dose, but it's still given, to what we'd regarded as a higher dose. So again, looking at the difference to see, is there any difference? So can you give a lower dose effectively and still have as good results or better results? And then as part of this study, we're also looking for certain signal markers in the blood tumor and imaging in patients who do less well, and potentially if we can predict who they are, we could maybe improve their treatments by adding other treatments in. And we'll also look at imaging and compare different sorts of imaging to look at how tumor has spread. And then as part of all this, there's quality of life questionnaires to be either filled in by the patient, the guardian, or the parent, depending on the age of the patient, at regular intervals throughout the trial. And so we can see when we're given different treatments in a randomized study, you might show equal in terms of outcome, but the quality of life might be very different. And it's really important from patients when they've been involved and their carers in developing these studies. Quality of life is something that is often poorly measured and not considered when looking just at outcomes like, did it come back or not, or have you survived the tumor? So just to get a bit of numbers, so most patients got Ewing sarcoma. There's this cohort that's recognized called Ewings-like tumors, particularly B-core tumors, and they are the Ewings-like tumors, which are often treated exactly the same as Ewing, so they are permissive to be in the study. It's anticipated that there's a minimum of 900 patients will be required in the study, and of that, we would need about 300 patients for the first randomization. That's the metastatic patients. B1 and B2, I'll come to in a minute. They're the radiotherapy question. There's the numbers. And randomization C is about the maintenance. And the eligibility, which is mandatory at the first point of entry, but it doesn't have to be at diagnosis, is obviously you've got a Ewing sarcoma or a Ewings-like tumor, but negative for the standard EWS R1 Fly1 gene rearrangement. When I said we're permissive age, we have excluded patients less than 2 years of age, but they're very, very rare patients. And obviously the usual written consent, and we're excluding anyone with a previous malignancy, because some patients can get Ewing sarcoma as a second malignancy. So this just gives you a trial schema. So effectively, you've got study entry. We'd like everybody to register at the beginning, so we get their data and collect biological samples. If you've got localized disease, you're not eligible for randomization A, just mainly because felt that their outcomes currently did not deserve adding in potentially another therapy which may have added toxicity. So that's randomization A, and we've just more or less completed what's called a phase 1B study, where we're combining regorafenib with the standard chemotherapy. And surprisingly, because a lot of people felt that regorafenib, which is a tyrosine kinase inhibitor, it would be too toxic to give alongside chemotherapy. We've demonstrated you can do that. And then when you come to having your local radiotherapy, if you require it, so if you want definitive radiotherapy, that is comparing 54 gray versus 64.8. So a lot of patients receive 54 as standard, but you can tolerate up to 65 gray. So we're hoping that will improve outcomes. And this is just about to open in the UK. A lot of these very big, complicated trials, there's often a stage opening. The B2 radiotherapy randomization is where, for whatever reason, you're giving post-op radiotherapy. Some patients, particularly younger patients, are being given 45 at the moment, as opposed to what might be regarded as the standard 54. So we wanted to compare those doses. And the most important thing about these questions, it'll hopefully demonstrate which dose is required for the best outcomes, not just outcomes such as reducing relapse and survival, improving survival, but of course, just side effects, toxicity, and quality of life. But for the first time in a randomized study, it'll give you data to support the doses that we should be giving as standard of care. And randomization C, which is maintenance. So standard of care is finishing treatment at the end of your 14 cycles. And then you'll be randomized or not if you want to take part in the study to vinorelbine cyclophosphamide for 6 months. Vinorelbine is oral and cyclophosphamide will be oral. And I'll give you some more details of that as we go through. So this just gives you an idea of the chemotherapy randomizations. This should open in about, I probably think, the third quarter of this year. You're going through all the usual regulatory hoops at the moment. So standard of care VDCIE, or after 9 cycles VCIE, and then you get regorafenib. And for the first few days of, if you like, the cycle of those 2 courses, you don't receive the regorafenib, but once you've had that chemotherapy, you then receive it, and you receive it alongside IE chemotherapy. And we have demonstrated in the phase 1b that this is tolerable. And then the same, you carry on to consolidation, although some patients will come out this side, they won't receive regorafenib and they get high dose therapy, but they'll have had it up to then. And then the other randomization is stopping treatment as standard of care versus 6 cycles of maintenance. So just explained, so this was the first randomization. This, as I say, should open Q3, and this is about adding regorafenib onto standard chemotherapy and hoping to improve outcomes. B1 and B2, so this is in active setup, and I'm not sure if you're aware, certainly in the UK, lots of Ewing sarcoma patients get proton radiotherapy. That's kind of standard of care paid for by the health service, unless you've got widely metastatic disease. And there are 2 proton centres for these patients in the UK. One happens to be in Manchester, where I am, and the other one is in London. So actually, it's quite, because most patients go into 2 centres, it's quite an easier thing to set up when you're not dealing with 20-odd centres that deliver radiotherapy. So again, that should open quite soon. And that's, as I say, the dose compared. And we have this organisation called Quartet. So basically, each plan will be validated and reviewed. So there's a standardization to the plan of radiotherapy for all patients on the study. And then the maintenance, this just gives you an illustration. Each cycle is a month. So you get weekly for 3 weeks oral vinorelbine. It's a soft capsule, so most patients can tolerate it, except for very small children or some people that won't take it, but you can substitute that for IV. And at the same time, you take cyclophosphamide continuously. And then there are rules about stopping it depending on counts. But the idea is that the majority of the patients that are on this maintenance therapy, which is a standard treatment we also use in another sarcoma you see in children and young people, rhabdomyosarcoma, it's very well tolerated. The aim is not to have low bloods. So most of these patients get back into normal life while they're on this maintenance. And there's various inclusion criteria around baseline testing. So this trial was developed through the Ewings consortium. And also we have a large international patient advocate group called Spain and the Sarcoma Patient Euro Network. And as a result, they actively took part right from the very beginning in the workshops to develop this study. And important things for patients and study was new treatment, because you look at those drugs that we use, they've been around since I started in oncology, even if we've tweaked with the recipe, as I call it. So they were heavily involved in designing the research and the early protocol, and particularly it's really important when you're trying to do patient information sheets and things, that they're involved in that. And they're also in the trial management group and the trial steering group. They were also involved in development both of sarcoma-specific quality life measure as for peds and older patients, and obviously the standard quality life measures, and also about how important it was that their samples are collected as part of the study, and that if you ask most patients and their families, they don't say no to samples. You always get some people who don't want to give samples for whatever reason, but it's the fact that they're often not asked, and obviously maximizing. So from the last study, a lot of the samples have been used not just by the original researchers who had biological questions in the study, but also other researchers as well. So they become a very valuable source. So far, we opened over a year ago. As you can imagine, small numbers initially as sites start to open. We've got 23 centres open to recruitment. We've enrolled 59 patients. Europe isn't open yet. Europe loves bureaucracy, and so we have to go through this whole new system. And of course, the UK is no longer in Europe. We're Brexit. So it's a separate thing because we're running the study and sponsoring it. So that's why there looks like a gap, because by now you would have expected some of the European centres, but that should happen quite soon. 59 patients are recruited and 24 have been randomised to arm C. And that's not bad, really. You don't expect more than 60% of patients to be recruited when you're doing a question at the end of what they regard as standard treatment. And so we should have 2 continents, 13 countries and 160 sites if it all goes to plan. And then I talked about sub-studies. I've talked about quality of life at various time points. PEDS, QUAL and European or ERTC, they're pretty standard. And then this is a sarcoma assessment measure that's been developed in the UK. And they're all into different languages. So that's important, obviously, in Europe. Biological studies sees how we take samples at the beginning and during chemotherapy. Lots of questions around trying to find, if you like, the whole thing around minimal residual disease, circulating tumor products, ctDNA, and hoping then to be able to use that to assess response, and therefore particularly, does it appear before patients relapse either clinically or on imaging. And then radiological sub-studies. It was, when we started the study, pretty standard to do bone marrow tests and aspirates to assess bone marrow involvement. But since the advent of FDG PET, some people don't do bone marrows anymore, they just do PET. But for those who still do both, we're going to compare and see if we can confirm prospectively with larger numbers that PET is what you need, and you don't need to do bone marrow aspirates and trophine, which is good. And the other thing is whole body MRI scanning better than FDG PET? So there's a question around that. And also using PET to see, does it reflect response? So after this induction block, we tend to do surgery if we can do it. And we use necrosis as a defining of poor response or good response, depending on the percentage of the tumour cells that are dead. That's very much a European phenomena. So we're going to use PET scanning to see, which a lot of patients would be having anyway, to see if that can be matched with other response methods and therefore predicting outcomes. So these studies are open, and then the imaging study should open quite shortly as well. So in summary, we should open randomization this year. A, B and 1 is very soon to be opened, certainly in the UK. We're currently going through the other layer of bureaucracy to open all the European sites. Randomization C is open. It's already open in Australia and New Zealand because they're not Europe. It's much, much easier. And this really is important because it builds on the success of 2012, which defined internationally, not just in states or related countries, that VDCIE was the chemotherapy we should do it. And actually, if you're wanting to add in treatment, it is much easier to add in treatment to that than other sorts of combinations. And it's designed as well to ensure equal access to the best available treatments and care, while creating a pathway for safe evaluation and introduction of future innovative therapies. Thank you.
Great. Thank you, Dr. Brennan. That was,
I'll
there's
stop
a lot.
A lot there.
There's a lot in there.
So it's
And,
a lot to present.
yeah. And everyone, as a reminder, keep just sending those questions through the Q&A box, and Piero and I will work through them with Dr. Brennan. So talk a little bit more about circular tumor DNA and how important is it? How important is circular tumor cells test after patient does the standard of care radiation or gets NED scans?
Yeah. So there's a lot of debate about detecting relapse, and many patients or families will know that they go back for repeat imaging, and it varies from country to country, and again resources, because MR scanning is not a cheap little test. And we also know that if you relapse and your relapse is more than 2 years out from treatment, you do better than if you relapse early. So is that just because by the time you pick up that relapse, it's usually clinically present? So you've either got symptoms, or you've got symptoms or you're not quite right, and you do some imaging. Actually, most relapses are probably not picked up on routine imaging, though lots of patients have it. So the idea from circulating, we know that we can measure circulating tumor products, and there's lots of different ways of doing it, and there's a lot of debate amongst the scientists which is the best way. And we have they've looked at small series, sometimes within the context of a trial, but ideally what you want to do is to be able to measure it during a study prospectively, and then look particularly as well that at diagnosis, does the burden of your tumor, so the amount of circulating tumor product, equal with your disease status, whether it's metastases or volume? We know volume is an important prognostic factor of your primary tumor. And what does it mean if it disappears? And if it disappears quickly, is that a good prognostic factor? So your effective treatment. And of course, if you then prove you can use it, then measuring it when you're on follow-up. And if you pick up very small amount of disease, so you pick up from a circulating tumor cells, and when you go to do imaging, you either pick up a small volume relapse or fewer sites, is that important, and does that alter your prognosis? Or does it mean that even before you can see something on imaging, you can intervene with treatment, and do you get better outcomes? And there's loads and loads of stuff being published on it, but no one has really done that kind of detailed, or the samples are incomplete in a large study. So even if not every patient has samples, if a large proportion of what we hope is 900 patients have samples and prospective samples, we hopefully will learn a bit more going forward. And if you have leukemia, you have something called minimal residual disease. So they measure again the amount of leukemia DNA, and that we know it's a standard of care test, and that predicts your treatment and your outcome. So patients' treatment will be changed. So that's the idea of hopefully doing this. And I think, yes, it is important, but it's about getting samples on everybody, and about the scientists really agreeing what the results mean, but also what's the right sample to use.
Great.
Following up on that question, is this trial set up to intervene early if you do detect something in the circulating tumor cells?
No, not at the moment, except that we don't know, first of all, definitely what it means. So we know we can measure it, and there's been smaller studies, so we want to see what it means. And then going forward, we also have a European relapse study called RECUR. And a lot of the patients who are in the upfront studies then end up in the RECUR study, because unfortunately patients relapse. And therefore, what we don't know is that if they end up in RECUR, and again, you picked up their recurrence earlier, inverted comma, which should give it a poorer prognosis, does it actually mean that when you pick it up in a small volume? And then you can continue to follow in the RECUR study. But we definitely don't know. And while we have drugs added in to Ewing sarcoma, so we've got regorafenib, it's really, really, really hard to get new drugs into Ewings, and a lot of drugs have failed. And certainly there is no wonder drug against the biological target of the translocation.
Yeah. Well, that's a perfect segue. I wanted to talk about arm A and regorafenib. what is the preclinical justification? It sounds like you've also maybe done
Yeah,
some
I haven't
trials with this
presented that.
Abesacro.
So yeah, so there's been lots of studies of single agent tyrosine kinase inhibitors in Ewing. So
Mm,
regorafenib was one. That study was called Regabone. There was a study with cabozantinib, again in Ewings. That was Cabobone, I think. So they're great, very unique
yeah, very much so.
name. And in Recur at the moment, the question is around lenvatinib, which is another tyrosine kinase. Again, seeing if that added into standard chemotherapy at relapse. So there's quite a lot. There's quite a lot of preclinical data. There's lots of preclinical data with chemotherapy showing synergism. So that's the reason to give it up front rather than as a maintenance, which some studies have looked at. And then there's lots of studies in relapsed patients where they've received single agent tyrosine kinase inhibitor and had some good responses. So there's enough evidence, we think. But
Hmm.
when we were looking for a new drug, and the best data out there in terms of preclinical data and other sarcomas with chemotherapy, so given to real patients as opposed to preclinical models,
Right.
and availability was in the tyrosine kinase inhibitors. And why did we end up with regorafenib? Because the company said yes, and it's as simple as that. And that's how the real world works. So there's lots and lots of preclinical studies going on that sometimes end up in early phase studies. But the really, really hard thing is to get drugs into large phase 3 studies. And this is probably one of the first studies in Ewings where you've added and it's a multiple target. It's not just one target. It's not specific just to one
Hmm.
target in Ewings, but it's multiple targets, but not conventional chemotherapy. So this is probably one of the first studies where you're adding it in up front. And we've chosen, certainly in the early study where we did it for toxicity, what's the dose we could give? We only recruited patients who had widely metastatic disease because they had the most gain with a new treatment and their outcome is so poor. And then for patients who've got metastatic disease, even though that could be just lung, which not bad compared to widely metastatic, which is the worst outcome, they're the ones in
Hmm.
randomization A. And the other thing as well is where you have patients who have lots of events to try and show improvement, your numbers for the study are much smaller than if you have some patients that are going to do really well. You need many more patients, to answer the question.
Yeah. Well, I want to commend you guys for trying something new, especially for advanced disease, because
Mm.
that's such an unmet need, as you've alluded to over and
Yeah.
over. So thank you.
Speaking of the volume of patients that you need for this, could you talk a little bit about the timeline for this? How long you
Yeah.
think it will take to have the study fully enrolled, and when you could expect to see some preliminary results?
So looking at the numbers, and particularly because other countries have joined, so particularly the bigger European country, Italy, and then there are smaller countries that have joined, but also Australia and New Zealand, which aren't massively populated, actually. They're big countries, but their population is nothing like the density in Europe. So in the last study that we did, which we actually closed it early before the number we thought we would recruit, we recruited it in less than 5 years with less countries, 640 patients in the randomization. So now we're looking at overall 900, assuming that even if you register at the beginning, you're not going to either be eligible for all randomizations or things. So I think the numbers would be feasible, but realistically, the randomization A, we have a bit of a limit on that and time because the patent for the company that has regorafenib goes. So they'll only give us a window of time where they'll give us the drug. So that's the deciding force there. But the other studies, we should have answered these questions, we hope, within the next 5 years, and some maybe sooner, depending on recruitment.
Okay. I'm going to pivot to arm
But that's a
C.
long time in patient lifetime.
Yeah,
It
absolutely.
is.
Yeah.
Okay. Thank you for that. I'm going to pivot to arm C, maintenance. We talk about maintenance all the times at Little Warrior and Ewings U. And just wanted to understand a little bit more about the regimen, how you guys came about that regimen, and why you think it could
Yeah.
possibly affect it for the patient community.
So maintenance was a discussion, and so there were discussions around, do you use a drug like, if you're going to use tyrosine kinase inhibitors up front, do you use a drug like that or similar one, as maintenance? It has to be a randomized study. It has to be comparing standard care, which is no further treatment after the end of 14. So those things were straightforward, and it was mainly pinching other people's ideas, as it were. So the rhabdomyosarcoma community, and that the FAR RMS study currently, but it previously was RMS 2005 that looked at adding the same regimen of maintenance, which had been used in a relapse setting in various tumours, including Ewings. And basically, that regimen was tried and tested. We knew the toxicity. And in the rhabdomyosarcoma population, so you can argue, well, it's a different tumour, they're sarcomas. We actually use similar sort of drugs, alkylating agents, alkaloids for those patients. And basically, patients that had received maintenance, there was nearly a 10% survival advantage for having it versus not. So we took a regimen we knew how to give. We had a bit of data to show it was effective. And we said, well, it would also seem to work quite well in Ewings and in rhabdomyosarcoma. So that's where that idea came from. Mainly because as well, we didn't have, and also because of its lack of toxicity and the fact that you can have a fairly good quality life on it. We know that from the rhabdomyosarcoma community. We also know that you're potentially, if it works, giving something that is not perceived as being difficult. Whereas if you start to use other drugs, potentially in the maintenance setting, and if they've got a lot of toxicity, then either people drop out of the study or won't go in the study. And you have to think as well about the age of most patients. So the average age is a teenager. And so when other studies have been done in teenagers, they're much, much harder to recruit because they don't want any more treatment. And that's different, if you're talking to a parent of a younger child. Most parents are quite willing to try anything to try and improve outcome. And in fact, some of the patients so far that I've recruited are disappointed when they don't get randomized to it, of course. But
I'm sure.
You can't give something that you don't know doesn't work.
Right. Thanks.
Great. There is a question here about eligibility and asking you to clarify a little bit more on newly diagnosed. This person asked, is a diagnosis 2 years ago classified as new?
No, we didn't actually. Traditionally, trials have had things like so many days from biopsy. So it is a patient who's just been diagnosed with Ewing's sarcoma. And most patients with Ewing's sarcoma would receive, from the pathway of starting investigations, biopsy to treatment, it's usually around about 4 weeks, maximum 5 weeks, depends, which country you're from and all those kind of things. So it is literally newly diagnosed and you've not received treatment. However, when we opened the trial, there were patients who, as standard of care, were having VDCIE chemotherapy. And when we opened the study, they were coming towards the end of their 14 cycles. So some patients were then recruited onto randomization C because they'd followed a standard pathway of treatment. So that's what I mean by newly diagnosed. But what you're not wanting is patients who, for example, have had either different treatment or they were treated and they're now a year out. Because on average, most treatment, if you just follow standard of care, is usually done within about 7-8 months of diagnosis maximum. So if they're 2 years since they were diagnosed, potentially, either if they're wanting more treatment, they're in a recurrence position or they've got new disease. Yeah.
Understood.
Cool. We're going to pivot here. We got a question following up on a comment you made about in Europe that you follow tumor necrosis a little bit more closely than the States. They were wanting to know, do you change frontline treatment as a function of having low tumor necrosis? If so, what does that look like?
We've always used a standard in the context of clinical trials where when they've had surgery, which we do after 9 cycles, which it's done earlier in the States, and we measure what percentage of the tumor is dead. And so if it's 90% or more, that's good tumor necrosis. If it's less than 90%, that's poor tumor necrosis. So certainly if you've got poor tumor necrosis in your, and this is your local, your localized disease, and you've had to have surgery, for example, and if you can't have surgery, just you can't assess it. In that scenario, you would add in treatment like radiotherapy. And rightly or wrongly, we use a lot more radiotherapy in Europe than you do compared to the States in terms of, after surgery, what percentage of radiotherapy. It doesn't mean people don't have radiotherapy, because there's still probably a population of patients around 30% who will have definitive radiotherapy, only they can't have surgery because site of tumor usually. So we change treatment in that way, or we add treatment in, but we haven't ever done a study other than high dose chemotherapy. And we showed in one of the really old studies, but there was lots of debate about it, and there was lots of editorials written about it by American clinical trialists about a European study, about where we did a comparison of high dose chemotherapy versus no high dose chemotherapy, just carrying on standard treatment if you had poorest local. So and there was some advantage, but the patients who receive radiotherapy as well, when they had poor necrosis, to receive high dose therapy, they had to have the tumor at a certain site. So you can't do high dose therapy and give radiotherapy to the spine, for example. And so you would choose radiotherapy over the spine versus trying to randomize them. And so for some patients, their therapy is changed. They have high dose therapy, but there aren't 100% believers in that, because as ever, there's lots of debate despite the randomized study.
Yeah. Can I just ask a follow-up question? When you say high dose chemotherapy, Bumeld, I mean, that's always followed up by a stem cell transplant, correct?
Yeah, it's that kind of that's what I mean by it. Yeah. And it's always been sulfur melphalan.
Okay.
Great. I'm going to talk more about the tissue samples that you're collecting. In the States, we have challenges where at that confirmation biopsy, the tissue isn't being thoroughly sequenced, and it's a missed opportunity often. Can you talk about within this trial and within kind of the UK generally, the attitude towards sequencing and what level of sequencing is done as part of this?
So, I mean, as you probably know, one of the problems or the difficulties about Ewing sarcoma is it's a very quiet tumor in terms of genetic abnormalities. In fact, 85% will have EWS FLI1, and that's it. And then there's some very rare partners with EWS FLI1 like ETV1, which will be in a tiny percentage, of patients. So generally, they have most patients will have next generation sequencing, but very rarely do you pick up anything other than what you expect to find. Other than if it's not a Ewing sarcoma, you might pick up a Ewings-like sarcoma, which has been diagnosed as Ewing's. In the UK, but not throughout Europe, there is a standard service offered to all tumours, certainly in childhood and teenagers, and in sarcomas, whole genome sequencing. But that has to be done on frozen tissue. It can't be done on paraffin. Obviously, most patients have a biopsy at diagnosis. So in theory, there should be adequate material. And as a result of that, so patients have gone for that. But again, it's a very quiet tumor. So it's at the moment, it's not that important, because actually for the majority of patients, you're not going to pick anything up other than what you know from standard of care testing, just using something FISH or something similar, a panel looking for mutations. So so that's how it works. But in other countries, it's not standard of care, and it is expensive. And it does rely on having frozen tissue. In the UK, all bone tumors, of which, if Ewing sarcoma, particularly if it's in a limb or trunk a bit, as opposed to, you will get some Ewings, in the jaw and things. they go to one of really 4 bone tumor centres in the UK. So they only get biopsied in limited centres, which means that there's a very good setup for tumour storage and consent, and getting consent for whole genome sequencing.
Interesting. Thank you.
I got a question about communication, since this is really a clinical trial for newly diagnosed patients. How do you, in Europe, UK, and Australia, et cetera, like, how do you educate the first-time patients that this is even an option? Is it really just getting buy-in from the doctors and the treating centers to start enrollment?
Well, I don't know what the setup is, you know. I mean, I know some of the setup in the States, but certainly in Europe, most European countries. So what happens is, if you have a suspected bone tumor, let's just talk about UK, which is easier, but it's very similar in a lot of other European countries. You don't just go to your local hospital who are orthopedics and get a biopsy done there. They go to one specialized bone tumor center that do all the biopsies and then do all the critical surgery. So all the limb preserving surgery was all developed in these centers, you know, so you're not doing amputations, and they've developed new operations. So you can do surgery in what would have been regarded, you know, some pelvic tumors are regarded as difficult tumors to remove. And even spinal surgery, which when I first started, very few people, they just got radiotherapy, but more patients get spinal surgery as well for spinal Ewings. And when the biopsy is confirmed, it can only go to several centres in the UK that only they treat Ewing sarcoma, and indeed other, certainly paediatric tumours, but also TYA tumours. So you're going to a centre, and all those centres open the study. So bar some emergency or some problem, whatever, it's Christmas, I don't know, all patients would be offered the study. So if they don't get offered it, or they don't go on the study, that's often their choice. And even if they had to have emergency treatment for Ewing sarcoma, they can still be offered the study. So, and it's very similar in Europe. Some of the countries less similar, like some countries like Portugal, it's less, but most patients go to a dedicated sarcoma center, and only they. And we don't have, I don't know what happens in, you know, in the States. So for example, because a lot of healthcare, I presume, is private as well, or people have insurance. So in the UK, you know, there are some of the centres that do private work, but that's mainly overseas patients coming to places like Great Ormond Street in London, that kind of thing.
Cool.
So that's why it's easier. So it's kind of, it's pretty standard.
Right.
I cover the northwest of England. So all patients with Ewings under 16 come to my centre, and they will all be offered the study. Yeah.
It's drastically different than here in the States.
Yeah, very.
So even though you've got a much bigger population, you've got much more buy-in from pharma in terms of getting new drugs.
Right.
Exactly. You know, it's much easier to recruit, despite the fact that the population overall potentially is smaller.
Great. We have a question about the side effects of regorafenib. What have you been seeing, or what would you anticipate?
Yeah, so there were a lot of, it was obviously first used in adults in other tumours, and it was perceived that it had a lot of side effects around potentially hypertension, so high blood pressure, some renal impairment or protein loss in your urine, etc., and abnormalities of your liver function. But in fact, it's been used in other pediatric studies, and in fact, certainly in younger patients who don't have potentially other morbidities, whereas if you're a sort of 60, 70-year-old and you've got another tumour, you've got other morbidity alongside. So most patients that come don't have any other problem. They're medically well and fit because of their age. So far, yes, you can get side effects with it, but it is actually really, really well tolerated. And we were really surprised when we did the early phase 1b study where we were giving it at a small dose. We went straight up to the top dose. Within 9 patients, we'd established the dose, and there was minimal side effects. So it's not, it can have side effects, and sometimes you have to either to reduce the dose or even stop it, but it's much better tolerated than I think people anticipated. And particularly when you're discussing, because it's an all-age study, you've got adult oncologists who use it in older patients for other reasons. So obviously they're a bit more scared about it and thought it would be too toxic, but it wasn't.
Okay, I got one more question here. Since we talked about arm A and arm C, I think it's only fair that we talk about B, which is the radiation arm.
Mm-hmm.
Is that only proton radiation, or is
no? The point I was making about proton, which hopefully is why it's easier to do this study in the UK, is that a lot of patients with localized or even with lung mets, because of the site of the tumor, they're the group that are eligible for proton radiotherapy. So they can have photon radiotherapy, which is the standard radiotherapy, and in fact, some patients will only receive that. But because of the service and how it was set up in the UK, then an awful lot of Ewing's patients who need radiotherapy are eligible for proton, and that's only done at 2 centres, and that includes the Republic of Ireland, Scotland, Wales, and England, and there's 2 centres for that. So one in the north, Manchester, and one in London down southeast. So it's not that they just can have proton, but it just so happens that a lot get proton. Whereas it's not necessarily the same throughout Europe, but it doesn't matter. There's no difference in the radiotherapy in terms of, if you like, tumor kill. The difference being for proton is you're treating less normal tissue.
Right.
So you should get less late effects and less long-term side effects.
But for unresectable lung mets, I mean, they're not part of this arm, right? You're not going to that high of dose to kill.
No, if you've got for whatever it doesn't matter whether or not you've got metastatic disease or not.
Yeah.
So, and for proton, lung metastases, as long as you've responded, are eligible in Ewings. Okay. Not just localized disease. But no, even if you've got metastatic disease and you're having definitive radiotherapy or even post-operative, that you are eligible to have that be in the radiotherapy question. So you might as well, you should include everybody that is eligible, and therefore establish, which dose is the best, and if it's the same, a smaller dose potentially has less side effects long term.
Okay.
And it's cheaper because you're given fractions of radiotherapy.
Yeah.
But health economics isn't really a question in it.
Oh, just better outcomes, right?
It, Yeah.
Yeah, absolutely. Emmy, do we have any more questions in the Q&A box?
Yeah, one super short question, and then one I'm just going to address. There were a few more questions that came in about ctDNA. We do have a Ewings U, a previous episode that talks all about ctDNA. So if you want to learn more about that, that would be a great one to zoom in on. One more question for you, Dr. Brennan. Someone is asking, is Royal Marsden Hospital participating?
Royal?
Yeah.
What was the name of the hospital?
It says, I don't see the Royal Marsden Hospital in London listed on your website. Are they signing up for this trial?
Yeah, they've signed up. Yeah, I don't think they're open now. Yeah.
Okay, great.
All right, we're going to bring this session to a close. Dr. Brennan, thank you so much for sharing your time and putting this trial together. Super comprehensive. And thank you for asking all the questions that you're asking, trying to find better outcomes for these kids. So thank you from the Ewing sarcoma community for being here with us today. Thank you to my fabulous co-host, co-founder Emily, for being here with me as well to moderate conversation. Lastly, thank you to our viewers for joining us here as well today. Please remember to stay up to date on future Ewings U episodes. The easiest way to do that is to subscribe to our Ewings U emails. You can go to our website, littlewarrior.org, navigate to the Ewings U tab on our website to sign up there. Also follow us on Instagram and Facebook, as we always post about our next speaker that way as well. Our next episode is already scheduled 2 weeks from now, Tuesday, May 20th, 11 a.m. Eastern. We're going to go to the NCI with Dr. Christine Hesk to discuss a relatively new clinical trial there, targeted therapy known as PEN 866. So the PEN 866 trial, May 20th, 11 a.m. Eastern. Please join us for that one. That's it for now. We'll see you here next time. Swords up, everyone. Thanks, Dr. Brennan.
Bye. Thank you for inviting me.
Absolute pleasure. Thank you. Bye.